Hello students, in this session of chapter number 12, that is biotechnology, we are going to learn regarding the second type of biological tool, that is cloning vectors. Now for recombinant DNA technology, agents are essential for the transfer of desired gene into the host cell. Such agents are called as cloning vectors, or they are also called as vehicle DNA. So cloning vectors means that these are the DNA molecules carrying a foreign DNA. Foreign DNA means the desired gene which we want to introduce in a suitable organism. So such foreign DNA segment and replicate in the host cell. That means the DNA molecule which carries the desired DNA and can replicate in the host cell is called as cloning vector. Then it is also called as vehicle DNA as it carries the desired gene. Now different types of cloning vectors can be used. For example, plasmids. Now plasmids are the extra chromosomal material found in some bacterial cells. Then bacteriophages. Now you know that bacteriophages are the viruses which cause diseases in bacteria. So such bacteriophages can be used as cloning vector. Then viruses like M13 lambda viruses are used as cloning vectors. Then cosmids, phagamids, then BAC. BAC means bacterial artificial chromosomes. Then YAC, that is yeast artificial chromosomes. Then different kinds of transposons. Then baculoviruses, MSC, that means mammalian artificial chromosomes can be used as cloning vector. Then the commonly used cloning vectors are mostly plasmids are used. Now the common plasmid which is used is PBr322. Now PBr322 is having or it is modified with marker genes. Then PUC and TI plasmid. TI plasmid is a tumor inducing plasmid found in Agrobacterium tumefaciens. So these are the different kinds of vectors which are used as cloning vectors, or they are also called as vehicle DNA. The most commonly used vectors are plasmids. Now plasmids are the extra chromosomal double stranded circular DNA found in bacterial cell, then they are found in yeast and animal cells. So these are the small circular extra chromosomal double stranded DNA present in bacterial, then yeast as well as some animal cells, and they are having the capacity to undergo the process of replication. Then the plasmid, when used as a vector, should be able to introduced in the host cell. Then it should have marker genes. Now marker genes are essential for different antibiotic resistance. Then it should have control elements, then promoter, operator and ribosome binding site. So all these characteristics are not found in plasmids. So these plasmids are constructed by inserting genes for antibiotic resistance. So commonly constructed plasmids which are used in recombinant DNA technology are like PBr322 and PBr320. So a plasmid should have all these characteristics when it is used as a cloning vector. Here we can see the plasmid a cloning vector. Now this is the plasmid, and this plasmid show ORI means a replication gene and a drug resistant gene, that is EMP, and this is the region where the foreign DNA can be inserted. So plasmids are used as cloning vectors. That means desired DNA is inserted in the plasmids, and these plasmids are introduced in a suitable host for multiplication of the desired gene. Then the vector used in plants is a TI plasmid. Now this TI plasmid is present in the bacteria called as Agrobacterium tumefaciens, and it is used for carrying genes in plants. Now this Agrobacterium tumefaciens, with the help of this TI plasmid, infects the plant, and the tumors are developed in plant due to this TI plasmid. So such TI plasmid can be used as vector for carrying genes in plants. Now this TI plasmid contain the transposon, that is tDNA, which inserts copies of itself into the chromosomes of infected plant cell. So such TI plasmid can insert its copies in the chromosome of plant cells. So it is used as a vector in plant cells. So a plant cell containing this DNA can be grown in culture medium or nutrient medium to form new transgenic plant. So with the help of this TI plasmid, the desired gene can be introduced in the plant cell, and the multiple copies of such plant cells are produced, and these cells can be grown into new plants, and as these new plants contain functional foreign gene or DNA, they are called as transgenic plants. So to modify the plant genotype, TI plasmids are used. The third biological tool is competent host. Now competent host means the recombinant DNA or the recombinant plasmid is introduced in a suitable organism or a suitable cell. So such cell or organism is called as competent host. The most commonly used organisms to produce the multiple copies of the recombinant DNA, or it is called as cloning, are bacteria. Now bacteria which are commonly used are Escherichia coli. So there is maximum use as a host of the bacteria, that is Escherichia coli. Then bacteria like Bacillus haemophilus, then Helicobacter pylori are also used as host for the transformation of recombinant DNA. Now we will see the topic, that is steps of recombinant DNA technology. Now you know that recombinant DNA means it is the DNA which has functional foreign gene or DNA. Such DNA is called as recombinant DNA, and this recombinant DNA can be introduced in a suitable host to introduce the desired gene or to make its multiple copies, that is gene cloning. So the first step in the recombinant DNA technology is the isolation of DNA from a suitable donor cell or donor organism. So in the first step, a DNA is isolated from the donor cell, that which type of genes are to be inserted in the host cell. So such cell which is selected to obtain the DNA is called as donor cell. And here we can see that donor cell, and then the DNA is isolated from the donor cell. So to isolate or to obtain the DNA from the donor cell, the donor cells are sheared with blender, that means they are broken, and for this purpose detergents are used. So after the breaking of the donor cell, the DNA is isolated, and such DNA which is isolated is treated with the enzyme that is restriction endonuclease. So due to the action of this restriction endonuclease, this DNA, that is target DNA, is broken down into small fragments of DNA, and then these small fragments of DNA are separated, then they are isolated, and then the desired fragment of DNA or desired gene is isolated. And similarly, a cloning vector is selected. Now the cloning vector may be plasmid. The most commonly used cloning vectors are the plasmid, that is PBR322, as it contains the desired genes, that is which are having antibiotic resistant genes. Then such PBR322 plasmid is commonly used. Then the vector plasmid is also treated with the enzyme, that is restriction enzyme. So as it is treated with this restriction enzyme, this vector DNA is cut at a suitable point. Then the desired fragment of DNA, which is purified from the DNA fragments, so such a desired gene or the target DNA is joined to the cleaved vector. Cleaved means the particular point where the plasmid DNA is cut. So at this point, the desired DNA is joined with the help of an enzyme called as DNA ligase. So this plasmid is inserted with the target DNA or desired gene, and these two are joined with the help of the enzyme called as DNA ligase. So this step is called as the insertion of desired gene in cloning vector. So such DNA in which the desired DNA or target DNA is inserted is called as recombinant DNA, or it is also called as chimeric DNA. Then the next step is transfer of recombinant DNA in a suitable host. The suitable host may be Escherichia coli. So most commonly used host to insert the recombinant DNA is Escherichia coli, or sometimes the bacteria called as Agrobacterium tumefaciens is also used. So to introduce this recombinant DNA into a suitable host, so here we can see a host bacteria, and in this host bacteria this recombinant DNA or chimeric DNA is introduced with the help of C++ ions. So such recombinant DNA is inserted in a suitable host, but sometimes different techniques can be used. That means there is no use of vector and the desired gene or DNA can be directly introduced in the host cell with the help of technique like microinjection, liposuction, then shotgun method. So, when the recombinant DNA is introduced or inserted in a suitable host, it is called as transformed host. Then after some time, the host cells are checked for bacterial transformation. Now transformation means the cells are checked that whether they have received this recombinant DNA or not. So the cells which have received this recombinant DNA are called as transformed cells, and such cells are selected, and this step is called as selection of transformed host cell. Then these transformed host cell are placed on a fresh nutrient medium for further multiplication. So the next step, that is multiplication of transformed host cell, occurs on a suitable fresh nutrient medium. So number of identical copies of this transformed host cells are produced by the multiplication. Then here we can see that the plasmid has undergone a replication. Then the multiplication of cell occurs, and you know that the gene expression occurs by the formation of particular proteins. So the gene carries its expression and produces certain types of protein inside this host cell, and here we can see that protein encoded, that means the proteins are produced which are encoded by the inserted DNA. So there is the expression of the desired DNA or the desired gene. So particular types of proteins are produced. So it is called as expression of gene, and so the expression of gene occurs to get certain products like different kinds of antibodies, then different kinds of proteins as well as antibiotics can be produced. So later on, if we need proteins, these proteins are obtained from the transformed cell, or this process is called as protein harvesting. Similarly, different kinds of enzymes as well as antibiotics can be produced by the desired gene or DNA. So in this way, the foreign DNA is inserted into a desired host, and the process is called as recombinant DNA technology. So recombinant DNA technology is used to get different important products like proteins, then different kinds of enzymes as well as antibiotics can be prepared by using this recombinant DNA technology. As well as recombinant DNA technology is also used to introduce the desired gene in certain plants.